In this tabletop exercise, we'll be utilizing Microreact. This is produced by the Virginia Department of Health. My name is Brianna Schoen, and I am a lab liaison at VDH.
And I'm James McCarty, a genomic epidemiology data scientist here at VDH. A little background for those of you unfamiliar, genomic epidemiology aims to understand the emergence and dissemination of high-risk clades within pathogen populations, with the ultimate goal of implementing evidence-based interventions to protect public health. High-risk clades are those subpopulations, strains, or variants of pathogens that carry risk elements, such as antibiotic resistance or virulence determinants. As a quick primer, here are a list of some common epi questions that genomics can help us answer, such as, does my locality have outbreak-associated cases? What are the genomic characteristics of the outbreak strain? What's the geographic spread of a cluster? What does the epi curve tell me about the outbreak? Is the outbreak ongoing? And how can I be alerted of possible new cases? The learning objectives for today's training include familiarizing yourself with Microreact, specifically how to manipulate project visualizations and how to add epidemiologic data to genomic result visualizations within Microreact, how to interpret genomic surveillance data and visualizations, including temporospatial and phylogenetic, how to apply genomic results to outbreak investigation, understand how surveillance data informs outbreak interpretation, and hypothesize about outbreak sources using epi-metadata and genomic results. Some things to keep in mind during this tutorial, you might want to have your Microreact icon sheet, exercise guide, and the answer sheet handy to follow along with this video tutorial. Today you will complete tasks to achieve 3 main goals by following the instructions on the slide while interacting with Microreact on your own laptop. Alternatively, participants may follow along with the trainer's visual demonstration of manipulating Microreact project panels and pre-built save views. As mentioned earlier, this tabletop will be comprised of 3 goals with 1 video per goal. With the first goal being to explore phylogenetic relationships and epi-visualizations for surveillance-level data. Next is to apply genomic results to outbreak investigations. And lastly, to understand how contextual surveillance data from ongoing regional genomic surveillance can help inform outbreak interpretation. Also, please note, this exercise is entirely a work of fiction. Any resemblance to a real-life situation is purely coincidental. We also want to cite our sources and acknowledge how this exercise was created by Sylvia Arjman and colleagues. Now, with that out of the way, let's jump right in with goal 1. We'll be using a pre-built Microreact project linked at the bottom of the slide here to gain experience with the software and interpreting genomic results. This surveillance level dataset has over 1,400 isolates sequenced from samples collected across the globe between 2013 and 2019. We're going to review the dataset and visualizations to examine the regional genomic epi of Club Pneumo in Colombia. Now you can optionally pause here and take a moment to explore the dataset and visualizations on your own before we get started with goal 1. A bit of background on our pathogen here. So Klebsiella pneumoniae, or Club Pneumo, is a gram-negative, non-motile, encapsulated rod-shaped bacterium. It is a lactose fermenter, a facultative anaerobe, and produces mucoid colonies on MacConkey agar. It is a commensal host of human intestines, so it's found in the stool, in addition to the nose, mouth, and throat. It's spread via contact with stool or wounds, and also historically improperly cleaned medical devices. It can cause pneumonia, meningitis, bloodstream and wound, or surgical site infections. Club pneumo is also associated with increased use of antimicrobials and immunosuppressive treatment. It is also one type of carbapenem-resistant Enterobacterales, or CRE, and here in the US, approximately 35% of CRE carry a gene for carbapenemases, which inactivate carbapenem and other beta-lactam antibiotics and spread rapidly among strains. So we'll go through this slide on what to do and then walk through the Microreact project together. So within each goal, we have a few tasks that are your action items. So feel free to pause the video at the end of this slide if you feel confident to try task 1 on your own. or keep the video playing and you can follow along with us. And it might be helpful if you have two screens if you're doing the latter. With all of that out of the way, let's dive into the tabletop with goal 1. First, we'll be exploring phylogenetic relationships and epi-visualizations for surveillance data. Using the Surveillance Club Pneumo Microreact project, understand antimicrobial resistance genes and sequence type data. And again, that project is linked at the bottom of the slide. Starting out, we want you to understand what map markers represent. To do so, we recommend using the ST resistance view. So once you're there, you'll want to change the color of the legend to location and modify the size of map markers to determine which location within Colombia appears to have the most cases in the project. If you'd like to try on your own, please pause now. Otherwise, we'll get right into the walkthrough.
So on our ST resistance view, we're going to click the eye icon at the top of the page. It's in the right-hand corner of the screen, and we can change the color column to location. This will change each isolate included in the project to be colored by the department or state they were sampled from. We can click on the legend to see the department corresponding with each map marker. Now we click on the configure icon on the upper right-hand corner of the map panel and click markers and click scale markers. This will allow the size of the markers to be scaled by the number of samples in each department in Colombia. By reviewing the scaled markers, determine which location, departmental or state, within Colombia appears to have the most cases in the metadata list of the Microreact project. And when doing this, you will be able to hover over each dot and see the number of rows. Each row represents a sample. So if we see in our metadata, each row is highlighted at the bottom here. And when you see the dot with 130 samples, click on it and it will be highlighted with a large circle. Looking below at the metadata, it will also check the selected isolates that are from this department. And when you look at the location column, we can see that the location with the most cases is Antioquia with 130 cases. So our answer for goal 1, task 1 is 130 cases in Antioquia. So now we're on to task 2. We're looking for the top 4 dominating sequence types in this project. And for this task, you should use the STs view. On to task 2. We're in the STs view, and we're looking for the top 4 dominating sequence types in this project. So we have this bar graph that displays all of the sequence types in all of Colombia. And when we look at this graph, it may be helpful to expand it. We can see that the top 4 dominating sequence types are ST11, ST152, ST258, and ST512. So our answers to goal 1, task 2 are sequence types 258, ST11, ST512, and ST152. On to the final task in goal 1. To discover the prevalent AMR mechanisms or AR genes detected, we're going to use the ST resistance view. So navigate to that view, and this time we're going to add new metadata blocks to the tree to help determine which AR genes are most frequently detected. And a little tip for those of you who aren't familiar with these genes, an easy way to identify them in the list is the 3 letters followed by an underscore with numbers. Those are the resistance genes. So we'll navigate to our ST resistance view, and we'll look at the phylogenetic tree. The bars to the right of the tree are called metadata blocks, and each yellow section of the bar represents the sample's detected AR gene. We want to make sure all the resistance genes are present by clicking on the configure icon on the upper right-hand portion of the tree. This is the configure icon. We want to select metadata blocks, and this brings down a drop-down menu. If you scroll down, we want to make sure all the resistance genes are checked. So the next question we want to ask is, are there any other rare or less prevalent carbapenemase genes in the metadata list of the Microreact project? And if we click on Legend, to the right here, it will pull up the Legend view, and any area marked in yellow, if we scroll down, represents a 1, meaning that the gene of interest was detected during the sequencing process. The green with 0 means that the gene was not detected. So which AR genes are the most frequently detected? All of the bars that have yellow portions within them are KPC2, KPC3, NDM1, CTX, M15, and just a few NDM9s, VIM2s, VIM24s, and VIM4 cases. So the answer to Goal 1, Task 3, is that the most common genes are KPC2, KPC3, NDM1, CTX, M15, and a few NDM9s and VIMs.